Hello, everybody. Thanks so much for joining us for our Zoom uh, discussion today. We're already starting the second month of 2022, and it's hard to believe, but I hope your year is off to a good start. And we want to thank you and your organization for your partnership with Research America in our alliance. And of course, if you're not a member, please get in touch and join us. I am really excited to welcome Stefano Bertuzzi, who's the CEO of the American Society for Microbiology. He's going to speak with us today about gain of function research of concern, which has been a controversial topic of late. And then we'll also discuss a range of topics with him, including COVID-19 sequencing and testing and improving pandemic preparedness, particularly around testing. So as always, please type your questions into the Q&A box or chat, and we will get to them at the end. Stefano, you were among our very first guests in the beginning of the pandemic when you joined former FDA Commissioner Mark McClellan and former CDC Director Julie Gerberding in April of 2020. So we are eager to hear what's on your mind now as you look in the rearview mirror and also as you peer ahead. So with that, let me turn it over to you.
Hi, good morning, everyone. And thank you, Jenny, for the introduction, and most of all for inviting me to speak with the Research America members. It's always truly an honor and a treat to interact with all the members and with the Research America leadership and staff. It's really a pleasure. So really, thank you. It's interesting you say that, right? In April, we were at the very beginning of this interesting journey that you all been in. And as you were speaking, I was thinking, we should go back and watch the recording of that and see what we said there, how much it materialized, and see whether what we were saying was of any help at all, and what we got right and what we got wrong. So I'll do that, actually. It's a good thing. Lots have happened here. And some really good things have happened, I think, from the research side. Obviously, the development of vaccines so rapidly and sequencing and many other things that we'll get a chance of talking. But also some really complex, confounding and concerning, I would say, things have happened in the space of research and policy. And one of these is certainly this focus in a highly political way on gain of function research of concern. And Jenny, I really applaud you for saying research of concern at the beginning, because this is going to be something that I try to emphasize. First of all, we don't particularly like the term gain of function research for reasons I'll explain in a second. But it's a term that in the policy development and the environment has been acquired and in a sense also imposed. And so we have to deal with it. And so expanding it to, given where we are now, to the explaining it further by adding of concern is really, really important. So if I can have the next slide, let's start right here with what is gain of function research. Essentially, it's a set of techniques and technologies in the lab that can give an organism, which can be any type of organism, a bacterium, virus, any microbe, but also vertebrates like mice, rats, birds, fish, and techniques that of genetic engineering that can let the organism acquire new properties or enhance existing one, or modify them in or modify certain characteristics of it. So that's the broadest definition of gain of function research. And if we move to the next slide, what I would like to emphasize here is a very, very important concept, is that gain of function research is not refer to pathogens per se at all. I myself, during my at the bench and in the lab research career, I had done a ton of gain of function research, like everybody who is in the molecular biology realm. And you can see I listed here few notable examples where gain of function research have been absolutely essential for research in cancer, cystic fibrosis, the development, actually one of the very first genetic engineering successes, which is a fascinating story for those of you who may have not had a chance of digging deep into this. There are a couple of books on the history of the discovery of insulin and how a devastating disease like diabetes have been completely transformed by gain of function research and being able to produce insulin, human insulin, and not using insulin from bovines that had a lot of side effects, and increasing the productivity to make a supply for people affected by this severe disease. So that's a prime example. And historically very, very relevant because it was one of the first genetic engineering application in biotechnology. But also in agriculture is very used. Resistance, for example, of crops like corn to insects that are particularly deleterious, could be controlled by that. And of course, the vaccines, including the COVID-19 vaccines, are the result of gain of function research. So I think it's clear that this is a very broad term that has very positive and non-scary use of for application in health, agriculture, energy, and whatever fields we can think. So in the next slide, you can go and see why are we then talking about it? Well, because the current framework, and I'll discuss that in a second, the framework for the preparedness for pandemic, potential pandemic pathogens, P3CO, is at the center stage of controversy at the moment. And this is essentially conflated with the problem of the origin of the SARS-CoV-2 pandemic. And there are members of Congress in particular who think that this framework under which the research infrastructure in the United States is operating was not effectively applied to certain grants. And there is a suspect that these grants could have been used for modifying the SARS-CoV-2 virus and being at the origin of the pandemic. Of course, let me be clear, there is absolutely no scientific evidence at the moment or seen in any form or shape that the result of the SARS-CoV-2 virus is of a gain of function, of a genetic engineering of the virus that created this gain of function. However, there is this, I would say, politicization of this issue, which has brought gain of function very at the center of this conversation. And to be clear, if we think about the origin of the pandemic, which is an issue that we don't exactly know how it started, and we wrote about this in several editorials and saying that, and ASM has taken some strong positions on this, saying, by all means, we need to do all the research that we need to do in order to understand what the origin of the pandemic is. It is very important without fear of retribution of where this may go. So like anything in research, we should be free to research without accusing or preventing open findings and open questions. But conceptually, we can think of essentially only two ways for the origin of the pandemic. And the origin of the pandemic and the origin of the virus are different things that I explain in a second. But for the pandemic, the origin can be by a natural spillover. So the virus moves from a natural reservoir in the woods, in the jungle, wherever, and spills over in the human environment. or it could come from a lab spillover, a virus that's being handled in the lab through a specimen that arrives there, or many other ways, could be leaked, could leak and enter the general environment. So these are essentially the only two ways by which a pandemic can start. Now, if we talk about the virus itself, there could be many, many ways by which a certain virus can become more infective, can more virulent, and et cetera, et cetera. And as I was mentioning, at the moment, there is no data at all pointing to the fact that gain of function research was actually, which in this case would be gain of function research of concern, was actually involved in the generation of the SARS-CoV-2 virus, which caused COVID-19. So because of all this, I use the slide to explain why this has been, to summarize things that you probably already know, but just to get us all on the same page. But I would try to avoid much as possible of using gain of function and origin of the pandemic in the same context, because really it can be manipulated politically with a small p, and on something that for which there is no scientific evidence at the moment. But, you know, these are the cards that we're given. And so it's important for us as a scientific community to actually be shaping the narrative in this. Otherwise, others will do it for us. And I think that that is something that is not desirable. In the next slide, and I won't go through all the bullets here, but I think the slide will be available on the Research America website, so you can look at this. I just listed here a long history and very relevant history of research oversight that the world and the United States have put in place to safeguard various aspects of danger coming from research. It's a long tradition that goes all the way back to the Geneva Convention in 1972, when there was a ban on biological and toxin weapons. Although in this particular case, it was research was not the focus of this, was just the use as of these organisms as weapons. But you can see here the list, and so it's very old issue. It's not something that's just popping up now. I have highlighted in bold the Asilomar Conference in 1975, because that is such a landmark for scientists who've been in molecular biology. And this was really a moment when genetic engineering became so common and so powerful. After the discovery of restriction enzymes, it became really immediately evident the power that we could do by cutting and pasting DNA together and modifying genes. And so the scientific community itself gathered at a conference center in Asilomar, decided to take a moratorium of these experiments. We need to first understand what are the risks that could be generated by this. And you see that this has progressed over the years. In the next slide, more specifically, I listed here what I think are the key events that are more specifically related to gain of function research of concern and where it comes from. I think in this particular area, the regulatory and oversight policy goes back to the anthrax attacks of 2001. And this really led the national academies to form a committee. There's been several committees, but the one that's been the most determinant has been the one led by Jerry Fink, called the Fink Committee, that really made recommendations for how to handle that research that could be concerning. And this led to the creation of the National and Science Advisory Board for Biosafety, which then got created in 2003 and is housed at the NIH, and has been dealing with many of these issues over time, starting in you remember the H5N1 flu outbreak and issues related to even before that with the publication of the sequence of the so-called Spanish flu. And so there were issues that they were very concerning and say, well, sharing this information could be put to bad use by some individuals. And some of those were actually overtly gain of function research of concerns. And so even here, there's been a moratorium on these issues, on saying we need to look into this before the government can fund research in this area. And this led to the framework that we're currently operating under, the 2017 that I mentioned at the beginning, the potential pandemic pathogens, or P3CL. This is now being questioned by some, and I think if it were not taken as a narrow political scope here, I think it could be a good idea to look at how this is working at a certain point, maybe once we are not in a crisis modality. And if there are any tweaks that need to be made, it could be looked at. I'm not against that in any way. This is my own personal opinion. But at the moment has served us well. And if you talk to the experts in biosafety and biosecurity, they actually think that it's a framework that has been, that served us well thus far. In the next slide, you can see that, the challenges that are underlying the problems that I describe is, as I mentioned, the origin of the pandemic. That's really the big one. But also these broader anti-China sentiments and this widespread sentiment of the unknown. And my concerns, honestly, here is that now the world gain of function will trigger what has happened with the genetically modified organism, right? Becomes the scary word, the bugaboo, something to be afraid of, because the issues are quite complex and hard for the general public to metabolize and fully understand. So this perception that the framework, was not properly applied and the lack of transparency or so, it is really dangerous, and it's something that is concerning. What is concerning is that actually it's leading what we're seeing coming from Congress, also a lot of very specific requests that go to the agency, NIH in particular, regarding specific grant. What are you doing here in this aim of this grant and here? And I think we're down the path of Congress micromanaging the individual grants, which is probably not the best thing to happen. Rather, we should be focusing on having guidelines and policies that are effective and making sure that they get applied in an effective way. So I think that with this environment, with the problems that I described, with all the underlying challenges that you see briefly listed in this slide, it's not surprising that there have been what I would call many legislative so-called opportunities for some to do something. And there's been really in Congress a sense of we need to jump on this. Many see this issue working with their constituencies. And so there's been several opportunities for inserting language in different bills that was to us, no pun, concerning. In the next slide, you see a list of the legislation that we've been tracking at ASM. And here, please allow me to give a shout out to the ASM policy team led by Alan Siegel and Mary Lee Watts, our federal affairs, who've been doing a terrific job in this space. And you see that the legislation I listed here, like the U.S. Competition and Innovation Act, USICA, that it originally contained some very troublesome language regarding gain of function. Fortunately, those amendments are not there anymore. They were not accepted by the House Rules Committee. We think this is very good news. However, now we're seeing language that mirrors what was in the DOE Office of Science Reauthorization that is problematic. So we hope that there will be room in reconciliation or so to still push for some modifications here. We're not completely satisfied with the language that is in there. Also, let me flag the Prevent Pandemic Act, and I saw that Research America has made a statement on that. It's excellent. And this is still the very, very beginning. And at the moment, we do not see language, but it's not impossible that there would be amendments or language appearing in the further, as the bill gets out of draft version in this front. So we're watching this very carefully because the concern that we have is that we could end up with language that could be very, very stifling of research at very, in a very broad way, with implications that may not have been considered before. As I said, we're all in favor of oversight and making sure that the research of concerns is addressed in a very effective way, and that scientists act responsibly, and the funding agencies and the regulatory agencies are doing their work here. Don't get me wrong on this. But we want to be very mindful of the unintended consequences of sweeping language that could have ramifications very important for the US research infrastructure. Because keep in mind that not all research happens in the US. And so if we're talking about competitiveness here, we also want to make sure that we don't end up with heavy burden of regulations and prohibitions that will really put our scientists in difficult circumstances, while other parts of the world could do essentially whatever they want. So that is really the concern that we have, and that's why we are looking at this very carefully. So in the next slide, what I have want to tell you a little bit what ASM is doing, and I'll be very brief here, but I'd like to mention, as I always like to do, is that there are issues. We don't do everything at ASM in advocacy. There are areas that we just follow. And since I'm speaking with the Research America crowd here, for example, appropriation research funding in general, it's something that we're very happy to follow Research America and our partners in here, because there's quintessentially nothing so specific about microbiology. I mean, there is, but we really think that we should all row in the same direction and advocate for the funding for science ultimately. There are areas that we just do not do. They're not up our alley or not our strategic directions. And then there are areas where at ASM in advocacy, we think we have the opportunity to lead because of the experts that we have among our members. And this is really this topic here of gain of function and dual use research of concern. This is a relatively small field in science, and it's a quintessentially microbiological problem. And ASM, among its members, all the leaders in the space are very engaged with ASM activities and leadership. And so I think we're well poised to work with everyone who is interested in this issue and provide our historical background in this and sources of expertise from our members. In the next slide, so with this in mind, you can see a list of things that we have been doing. And again, the policy team has done a terrific job here in meeting with staff and members, both in the House and in the Senate. And also a lot of activities ongoing with the agencies and with the Office of Science, Technology, and Policy. And we have also tried to amplify this message with the general public. At the end of last year, we have published an editorial in Stat with the former ASM president, Dr. Vic Derrida, and several other things in this area. The next slide, you can see other activities that we have in place, and I'd like to flag in particular the last bullet here, that tomorrow at 2 p.m. the team will hold a conference call with the several stakeholders. These are the legislative, government relations staff of various university and research center to have a conversation about this and bring them up to speed to what we're doing and what the issues are from our perspective, try to make sure that we coordinate as much as we can. Finally, if I can move to the next slide, I'd like to spend a word on publishing. ASM is a very large publisher in microbiology. We publish about 15% of the world share of microbiology articles and receive about 30% of the citations in microbiology. So we feel we have a particularly strong responsibility on the publishing side in ensuring that we have processes in place that are responsible for handling dual research of concerns issues and gain of function of concerns research. You see in the schematic what our process is. We start by asking authors to disclose if they think they have gain of function, dual use of research concern, or any P3 issues in there. Also, we don't just rely on the authors disclosing that, but we have ASM editorial staff with scientists that actually screens the manuscripts, exactly looking for this. And if there is something that is flagged as relevant for this topic, we will assign it to someone with high expertise in biosafety concerns as a special reviewer for those issues, so on top of the regular reviewers for the articles. And then eventually all the comments on this front will be relayed to the editor and the editors-in-chief that will work in thinking what they would like to do with this article, given the issues that have been raised. The editor-in-chief can, if the issue is particularly serious, convene what is called the ASM Responsible Publication Committee that is going to have experts in this area, and they may even seek further outside advice on how to handle the manuscript. And in the end, the recommendation could fall in different scenarios. It could be that the article is rejected. It hasn't happened so far that was the case. Or it could be redacted. Certain information may be taken out because it's considered dangerous or not appropriate for being published. It could be published as is, but maybe accompanied with an editorial that would explain why it's publishing and what was the process and why this is being published, or just published without any accompanying editorial in case the situation is particularly clear and simple and there's no need for that. So I just wanted to mention this to say that it's very important that when as publishers that we also keep this in mind, because it's a very big responsibility and we want to take it seriously to make sure that we have robust processes in place. And I think this is all I wanted to tell you today, and delighted to hear your questions or any other issues you'd like to talk. And I also would be very, very interested in discussing issues about sequencing in particular for this, because it's a very hot topic we're working on and connected to diagnostics and other issues. So we can take it at any time.
Terrific. Thank you so much, Stefano. I think you have a few more minutes, right? Because we're at our scheduled time, but I think we can go longer. Well, you have done a really terrific job putting gain of function research in its proper context scientifically, and also shared, the extensive oversight framework that exists around it. And you also spoke so clearly about the way that the origin of COVID-19 has been conflated with gain of function research. So just tremendously helpful. It's just one of many examples where scientific topics and scientific recommendations have become politicized, over the course of the pandemic. If there are folks on the line who may not have been invited to your call tomorrow, perhaps, Alan, we could put your email in the chat and folks could reach out to you. That would be great. And your point about needing to shape the narrative is so important, right? We've seen this again with social distancing, masking, vaccination, et cetera. I'm going to just take a few minutes to ask you about sequencing, because I know that the sequencing has been critical to our early identification of COVID-19. And since then, tell us about the progress that's been made, what we may be able to do differently at the beginning of the next pandemic, given some advancements in the sequencing arena.
Yeah, very, very important topic, Jenny, as you point out. Unfortunately, we had some catch up to do on the diagnostic side here on the sequencing. And I think something terrific, though, has happened on this front. And this has been, I really want to give a big shout out to the CDC for really taking a strong leadership on this front. They had a program, which was a very small program. The Advanced Molecular Detection Program was about a $30 million program when the pandemic hit. Now, this program has reached $1.75 billion. This has been a big infusion with the pandemic funds, and this has been terrific. And let me tell you, advocacy works. This has been a very big topic for advocacy for ASM and other organizations. And we're so happy to see that. So now we're through this and through the centers of excellence that CDC is working, is putting in place an infrastructure that is focused on creating a national network, ultimately, that would allow for capillary sequencing of variants. Because if we don't have a variant, if we don't have sequencing, we really are fighting, blindfolded, essentially. And I would like to make a point here that I think it's beyond surveillance. Surveillance is extremely important. But what I'm looking for is, and what I'm advocating for, is a holistic sequencing infrastructure in the country, because surveillance can happen in many different places. So, for example, the hospital labs are often sentinel labs for surveillance, because that's where the first cases pop up. And so if we have a system, an information system, a sequencing infrastructure, a spoken hub system where all this information is relayed, we would do such a better job to really creating that infrastructure that would be transformative. And I am so looking forward to envisioning what the clinical lab of the future will be, because the clinical lab of the future will look very, very different than the current clinical lab. And these funds, this infrastructure will be so enabling of this. I tell you, I could not be more excited by what I'm seeing here. We can really move the needle.
Stefano, that is great to hear. I think you're also talking about the CDC really empowering public health labs,
yes,
state public health labs around the country to have sequencing technology and the expertise, right, to play a role here. I'm going to turn this over to my colleague, Anna Platt, to see what questions we've gotten from our audience.
Absolutely. And as a reminder to folks, please feel free to put your questions into the Q&A or the chat, and we will do our best to pose them. So one question that has come in, as we look back on the pandemic, what, in your view, are some of the most important lessons we've learned, both positive and negative? And what have we learned about testing, including effectiveness and availability?
Yeah, thank you for this question. So I would say, in general, what we learned is that we need to act quickly. I think very often there's been hesitation, and this is worldwide, I think, in taking very big decisions because we're not sure. You had to make decisions with very limited information at the beginning. But viruses move really quickly. And so very often we put in place measure when it was too late. And so being able to have as much information early as possible, go back to what I was just saying about having an infrastructure of sequencing and integrated, that's essential for that, and making decision quickly. The second element is that I think will need to change and we need to learn for the future. And this is so important. And it's typically, as we all hear talking about research, is something that I don't think we have valued as much, is that of the supply chain. So typically as scientists, we don't spend much time in the shipping and receiving department of the university, right? And in reality, we realize with the pandemic how hard this has been. I'll give you this example, which works for everything, I think, in what I'm trying to say. At the beginning of the pandemic, when it hit, there was one company, Copan Diagnostics, that had the market share for 95% of the global share of nasopharyngeal swabs that were needed for testing for SARS-CoV-2. One company, 95% share. That company happened to be in Brescia, Italy, actually a few miles from where I grew up. And all of a sudden, the whole world needed this. But there's more. As they ramped up production to try to supply this essential tool, that's when all the flights were shut down. So there were no flights from Europe to the US, or very few, and loaded with cargo instead of passengers. And so there wasn't enough supply coming here. And so we worked very actively trying to make things out. We had a little bit of knowledge village there as well. And so there was a US Air Force base a few miles from the location of that company. And we're able to engineer 3 military flights a week, bringing over swabs, essentially. So that's not the way to handle a pandemic, right? This was a really a management by crisis. So really developing infrastructure that is capillary and ensuring strong supply chain, we learned it's so important. There will be many more things, but let me limit to this as I think a strong learned lesson, hard learned lesson.
Oh, looks like, oh, Jenny, you're muted.
Do we have additional questions, Anna?
We do indeed. So another question that has come in, let me just pull up the chat here. What advancements in diagnostic testing do you think will be most impactful for future pandemics and even smaller, perhaps more limited disease outbreaks?
Yeah, I would say, I know we're over time, but because I'd like to talk about this for like 15 hours, I won't, trust me. But I think the biggest thing here is rapid testing and point of care testing. That's clear. We need very reliable tests that can be done very, very quickly. And that's very important. So that to me is really what we have to work on. And then also the management of all this, because one thing that I'm really torn, ASM is all for testing. But what we see is that very often much of this testing has been, especially in the beginning or halfway, I would say, it's been clogging up the microbiology labs in the country. And even our people in the hospital say, we don't know what to do first. We need to take care of the very sick patients in the hospital. And at the same time, we have this extra load, let's say, from the university that needs to screen the students that are there. So really having this infrastructure that allow us for rapid, reliable testing and done in different settings and have a coordination for all this would be so important.
Well, we have clearly run out of time. And Stefano, I could talk with you about diagnostics for forever, but I have learned so much today, and I'm sure our audience has as well. So thank you so much.
Thank you.
And just a quick announcement before we end next week, a Thursday at 2:15, we're hosting Neil Lane, who is the Senior Fellow in Science and Technology Policy at Rice University's Baker Institute, and our 2022 Herb Pardis Family Award for National Leadership and Advocacy for Research honoree. So this is one of our special discussions in conjunction with our March 16th Advocacy Awards. And I'm really excited because one of our impressive early career scientists on our staff will be speaking with Dr. Lane, Victoria Schneider, and I know she's going to do a terrific job.